days ago, I accepted an injection of HPV vaccine. To be honest, it is the most painful injection I've ever had. I was told that it can prevent me from cervical cancer, skin warts, and anal cancer. Thus, I wonder what it contains in this 5 ml liquid. In order to know that, we're going to learn more about therapeutic cancer vaccines, about what are they, how they work, why I can now treat all the cancers, and who can benefit from them. The HPV vaccine I just took is called prophylactic cancer vaccines. It can kill carcinogenic virus like human papillomavirus or hepatitis B virus and prevent infections so cancers cannot be induced. Another type of vaccine is called therapeutic vaccine. It's for those who have cancer already. It targets at a certain proteins or antigens on cancer cells, which will be mentioned later, and thus triggers immune response to kill these cells. So generally, how these vaccines fight with cancer? By immune response. As the tumor antigens contained in the vaccines are injected, they are captured by antigen-presenting cells like dendritic cells through phagocytosis. APCs then migrate to lymph nodes and triggers activation of T cells and B cells. T cells are differentiated to memory T and effector T cells, while B cells are differentiated to plasma cells. Effector T cells can enter tumor microenvironments, recognize and engage cancer cells through T cell receptors. Thus, it can either release porphyrin and granzyme to perforate the cell membrane and cause direct cytotoxicity, or secrete IFN gamma and TNF alpha indirectly, cause cancer cells apoptosis in final. At the same time, plasma cells can produce antibodies bind specifically to antigens on cancer cells, and then recruit natural killer cells by their Fc receptors. Cancer cells will be killed by either cytotoxic granules or induce apoptosis. As antigens are so remarkable that triggers a series of immune response, you might be wondering which type of antigen is suitable to be used in vaccines? Well, there are two kinds of antigens on tumors: tumor-associated antigens and tumor-specific antigens. TAAs, such as melanoma antigen or prostate-specific antigens, are kinds of overexpressed proteins, mainly in tumor cells. It then triggers a series of antigen-presenting machineries, APM, which will be mentioned later on. However, it exists not only in malign cells but also in benign tissues with low expressions, which increase the risk of autoimmune toxicity. Also, due to the central immune tolerance of the thymus, activated T cells that recognize TAAs or other autoantigens may be eliminated during development, which will affect the efficacy of the vaccine. Many clinical trials targeting at TAAs have received disappointing results. For TSAs, you might heard some of them like human leukocyte antigens. They are also called new antigens. These antigens are individual-specific and non-autogenous, so the off-target damage is limited with no central immune tolerance. Compared with TAAs, new antigens have more potent immunogenicity and higher major histocompatibility complex affinity. With the wide application of next-generation sequencing, they are seen as the rising stars in recent researches. Knowing all these, are we able to make vaccines for all the cancers? Sadly, intrinsic and extrinsic immune resistance stops the development of new vaccines. The most common reason of resistance is immune selection due to the difference of immunogenicity between tumor cells. Those cells with strong immunogenicity can easily trigger anti-tumor immune response, while the weak ones can only escape. The process of natural selection will cause the continuous decrease in immunogenicity of cells. Apart from that, some cells are able to avoid T cell killing by downregulating expression of HLA on their surface and then continue their proliferation. Furthermore, changes in tumor intrinsic signaling pathways may also cause immune escape, like the activation of WNT or beta-catenin pathways. By designing better delivery platforms and improving combination therapies, we could try to avoid these complex mechanisms. Finally, 3 vaccines successfully passed the FDA reviews and got approved now. They are BCG vaccine, Syphilis-T, and T-VAC. They are the most lucky ones among all the 3,000 drugs in the clinical trials now. First, let's see how BCG vaccines fare with bladder cancers. As the vaccine was injected, it can be internalized. Cancer cells then upregulate expression of MHC2 and ICAM1 and secrete cytokines. Immune cells can be recruited after and cause cytotoxicity. Although it has been the standard treatment for non-muscle invasive bladder cancers, especially carcinoma in situ, its detailed mechanisms is still not clear. The patients often need to take 3 years injection after operation called transurethral resection of bladder tumor. Years ago, a vaccine based on oncolytic virus, the T-VAC, was approved. The viruses are genetically modified to express human granulocyte macrophage colony stimulating factors. T-VAC has a dual mechanism of action, an oncolytic effect whereby it directly infects and kills the local tumor cells at the injection site, as well as an immunotherapy effect through the induction of local and systematic immune responses. It causes release of a soluble tumor-associated antigens which are ingested by dendritic cells and then presented to CD4+ helper and CD8+ cytotoxic T cells. It's suitable for treating cutaneous melanoma, especially in head and neck. The overall response rate was about 35%, which is not high, and could cost up to $300,000. Until now, cancer vaccine research is undergoing an intense surge in early-stage clinical investigations globally. Next-generation sequencing, increased computing power, and advanced algorithms have significantly boosted our researches. We can expect more new antigens are identified, and the delivery platforms can be further optimized. However, we still need to surmount the immunosuppressive TMEs and reduce the cost of these therapies. If you want to know more about these contents, please check this reference page to get more info. These cancer vaccines are useful, but I hope that you will never need to use them. And that's it. We wish everyone peace and healthy every day. Bye bye.